Good afternoon, everyone. My name is David Jin, President and CEO of Avalon Globocare. First of all, I would like to thank Bernie Siegel and the Regenerative Medicine Foundation for giving us the opportunity to feature two of our cutting-edge technology platforms at this year's World Stem Cell Summit. Avalon Globocare is a NASDAQ-listed biotech company dedicated to develop and deliver innovative and transformative cellular technologies and therapeutics in the fields of immuno-oncology and regenerative medicine. In this session of Hot Topics and Future Trends, we are honored and privileged to have two world-renowned scientists as our speakers, and both of them are the principal investigators of Avalon's academic industry co-development projects. These are our two speakers today. Professor Uwe Sleytr, Professor Emeritus at the University of Natural Resources and Life Sciences in Vienna, Austria, is also a full member of the Austrian Academy of Sciences. Professor Sleytr is a pioneer in S-layer nanotechnology. Our second speaker today is Dr. Shuguang Zhang. He is currently the director of the Laboratory of Molecular Architecture at the Media Lab of MIT. He is a fellow of the United States National Academy of Inventors. He's also a recipient of the prestigious The Protein Society's Emil Thomas Kaiser Award in 2020. Dr. Zhang is the inventor of the QTY Protein Design Code. So I hope you enjoy these two special featured presentations brought to you by Avalon Globocare.
I'm going to talk about S-layer proteins as building blocks for nanobiotechnology applications. Our surface layers, the abbreviation is S-layers, are crystalline monomolecular protein or glycoprotein arrays, representing one of the most commonly observed surface structures in bacteria and archaea. And what can be seen here on the left and right panel are high-resolution electron micrographs of potentially living cells. We see the cell surface colored with a square lattice on the left and the hexagonal on the right side. And what we can also see in the middle is the types of S-layer lattices observed. We find oblique lattices composed of monomers or dimers. The morphological units are always shown in red. We have square lattices composed of tetramers and hexagonal arrays composed of trimers and hexamers. What we also can see from the schematic drawing that S-layers are porous structures. Now, I would like to stress at the beginning of my talk that S-layer proteins are one of the most abundant biopolymers on Earth. If we think about the biomass of eukaryotic organisms, they are roughly 1/3, and the biomass of prokaryotic organisms, they are 2/3. We just don't see them. And in prokaryotes, it's the bacteria and archaea. And the reason it took so long before this was recognized is the fact that the classical experimental strains in prokaryotic studies are Escherichia coli and Bacillus subtilis, and they don't have an S-layer. This is just by coincidence. S-layers make about 10% of the cell protein. Now, a few words about the function of S-layers. This is not a complete list, but in archaea, the S-layers are involved in determining the cell shape. S-layers act as molecular sieves and antifouling coatings, so the S-layers prevent molecules from entering the cell, but also from leaving the cell. They generate some sort of periplasmic space. They are protective coat. This can be phages, this can be these pathogenic organisms, phagocytosis, and so on. So several features which can be summarized under protective coat. They are also involved in surface recognition and cell adhesion to substrates. For instance, lactobacilli stick specifically to epithelial cells, and adhesion zones for exoenzymes. So the cells don't lose these molecules, they just stick to the surface when they degrade polymers. They act as virulence factor of pathogenic organisms, and the features affecting the hydrodynamic surface properties of S-layers Now, this is a very interesting area which we are studying in detail in the presently, but it's sort of a different world that goes into the biophysics. Now, S layer cover rounded surfaces. This can be seen on intact cells. This is a hexagonal array, and we see that S layers are a low free energy arrangement of molecules. We even see the lattice folds required in a hexagonal array as schematically shown here. The same you can find on buckyballs or fullerenes, where you have pentamers instead of hexamers. This is my favorite slide. It sums up in a very nice way the unique features of S layers if we think about them as patterning element for molecular, as a molecular construction kit, particularly in nanotechnology, nanobiotechnology. It's a picture from Escher. We modified it in some extent. If you look at the white lizards, they are S-layer lattices composed. We see that the S-layer lattices are composed of identical species of subunits. If you look at the black lizards, we can say pores passing through show identical size and morphology. And what we also see immediately is that functional groups are aligned in well-defined positions and show defined orientations. And this is a very important feature if we go for chemical and genetic modifications to change the hydrophilicity, the antifouling properties, the pore size, or the surface charge. We see immediately also that S layers are highly porous structures. In this case, it's with the white and the dark lizards, we can say the porosity is about 50%. An important point is the self-assembly, the reassembly of S layer proteins. If it goes also, this is important for the native cells, but also for the growing and dividing cell, but also for the different applications. We can isolate the S layer subunits from the cell surface, or we can use S layers which have been produced by in E. coli, for instance, recombinant products, the inclusion bodies. So the isolation is done by hydrogen bond breaking agents, guanidine hydrochloride or urea. And then the isolated subunits can reassemble, recrystallize. This can happen in solution. We get cylinders, flat sheets, or vesicles. But we can also crystallize them on liposomes, emulsomes, nanoparticles, nanocapsules. But an important point is also that they actually crystallize on interfaces, on surfaces. This can be the air-water interface. We can crystallize them on solid supports. This can be metals, polymers, semiconductors. We can self-assemble them on monolayers, which are generated on solid supports or on lipid films. The crystallization can be also studied by atomic force microscopy when it occurs on solid supports. Here we have a silicon wafer. We see the nucleation sites here, then the crystals grow until they meet each other, and finally we have the closed surface monolayer, as you see here. Of course, the different orientation of the crystallites is shown here, and this is at higher magnification. We see that these morphological units. S layer can cover our quite small tubes. In this case, we have carbon nanotubes, and they are covered here with, you see that the diameter is about 30 nanometer, and we completely get a coherent layer on such a fiber-like structure. So if we think about the nanobiotechnological applications of functional S-layer arrays, one of the first applications we developed was ultrafiltration membranes. We use them as matrix for a well-defined binding of functional molecules and nanoparticles. This is for surface patterning elements and supporting structure for functional lipid membranes. This can be planar membranes, liposomes, and emulsomes. We can also produce virus-like particles, which is interesting for delivery and targeting systems. We call that the Trojan horse particles. Now, Cellular ultrafiltration membranes, this was the first application. We studied the permeability properties on the intact cells, but what we found is immediately that Cellular's work in the ultrafiltration range, which is a cutoff, as I will show in the following. But the idea then was, how can we exploit a structure which has been optimized on a cell surface in a square micrometer? How can we produce square meters? And the idea then was like tiling a roof. We deposited the S layers on microfiltration membranes, as you see here, and this was under high pressure. This is the machine we developed. There's a fully automatic processor controlled apparatus, and we saw that this generates a coherent layer. This is a cross-linked protein later on. This can be, for instance, done with glutaldehyde, and we reduce the shift bases with borohydrate, and we get a very stable membrane. It's almost like a polyamide membrane. If you look at the ultrafiltration membranes, we have a sharp cutoff. What you can see here, this is the retention of a molecule in the supernatant during the filtration process, plotted against the molecular weight. And we see that the ovalbumin, the bovine serum albumin, is retained to about 100%. This is a 67,000. It's, by the way, very interesting because the cutoff is very similar to native kidneys, which is 65 kilodalton. The ovalbumin is retained to almost 95%, and the carbonic anhydrase, the 30,000, easily penetrates through the membrane. It's retained almost 30%. So this shows that apparently the molecule requires a certain orientation for passing through this pore. Please remember at this point the black lizards in the picture by Escher, and of course the smaller molecules easily penetrate. So the next application I would like to talk about is chimeric S-layer proteins. This is schematically drawn. The horses or the knights, if you look at chessboards, would be the functional part. This is an antibody, enzyme, any ligand, which can be bound to the S-layer, and the S-layer protein is responsible for the regular array. And what we see, so the horses in this case are really oriented in a well-defined position and orientation. This is an important advantage to the classical immobilization, which you can see here, the functionalized S-layer. If we bind molecules to a surface for generating functional surfaces, we use straightforward chemical methods. In most cases, we activate carboxyl groups with carbodiamide, and then these proteins, for instance, an enzyme or an antibody, will stick to the surface like insects sort of immobilized on a sticky tape. There are many areas where this orientation, the functional molecules are oriented in a way so the functionality is not active or not accessible for, let's say, binding another molecule. With S layers on the right-hand side, you can see that we have a defined orientation. All these functionalities in a defined orientation, and this is a great advantage. Also, if I think about signal-to-noise ratio and unspecific absorption, which definitely happens in such a case. Now, seeing is believing. I show one example, and this is streptavidin, which is quite common ligand used in different applications, diagnostics and affinity membranes. In this case, it's an oblique lattice, so a P1 lattice. This is a negative staining image. We see the pores. This, by the way, here are two types of pores, but again, we would have a very defined cutoff to the larger pores. The area indicates the area white is here where major masses are seen in the lattice. If we mobilize the streptavidin, we get a definitely increase in this area. And the interesting thing is, if we use a marker molecule biotinylated, like the ferritin molecule, also the ferritin binds according to this crystal lattice. So this is summed up in the schematic drawing. We have the S layer, we have the NITES, that would be the streptavidin, and the immobilized biotinylated ferritin. Oh, by the way, we also see that in atomic force microscopy. So this is just a few examples for S-layer fusion proteins. In the left, you see this abbreviation for the protein. These are S-layers from thermophilic Bacillus species. Here we have the length of the functionality, and here is the functionality, just why the reason why we use this recombinant fusion protein. For the core streptavidin, it's 118 amino acids. This is the example I've shown before, binding biotinylated molecule. This was an interesting example. We used S layers for immunological experiments with allergy, development of allergies. It's the PETV1, the major birch-borne allergen, streptag, a short amino acid, a short peptide, then peptide mimotopes, for instance, Epstein-Barr viruses, then the IgG binding domain from protein A, the same is also used for protein J, and different fluorescence proteins. These are quite large moieties, but they still easily crystallizes. Then for different enzymes, I just do the example of the thermophilic enzyme. And the interesting aspect, we can also fuse the heavy chains from chemo antibodies. This is just summarizing these different ways for generating functional S-layer protein, fusion proteins. This is the single functional S-layer protein. Here we have refolding. This would be the heterotetramer for the streptavidin, what I've shown before in more detail, and the multimeric enzymes where we immobilize the enzyme flexible link, the enzyme finds aggregates here in the on top of that. So I go now to the next application, and this is S-layer coated nano architectures. We see in freeze etching the vesicle here with the highly ordered structure, and this is the classical example how we coat magnetic beads. Just a mistake. We cut the magnetic bead. This is normally this beads are coated with a polyester shell. Then we have the S-layer fusion protein. This can be the ZZ domain here, the synthetic analog of the B domain for protein A, or it can be from the protein G. And this is just a comparison to show how efficient such a regular array can be. This is the percentage of the elution capacity, the removed antibody of a cell culture supernatant, where we edit these magnetic beads, and these are just commercially available beads. And here we see this is a considerable more binding and release later on with the S-layer system. In the following talk, Professor Shuguang Zhang will describe a very, very interesting approach for modifying the 7 hydrophobic alpha helices of transmembrane proteins, particularly the so-called G protein coupled receptors. And we combined our both technologies in that we edit to the G protein coupled receptor the IgG-Fc part. So we have this fusion protein, and we used the S-layer protein A or protein G for generating the patterning. And what you can see in the following is that in the schematic drawing that Professor Zhang will describe this in more detail, that we have a very efficient distribution of these transmembrane functions. This is, I think, a very, very interesting aspect. So the applications of S-layer fusion proteins summarized: we have a sensing layer for label-free detection systems. studies can be done with surface plasmon resonance, quartz crystal microbalance dissipation. We have high density affinity coatings for downstream processing or apheresis. We can use them as catalysts for if you mobilize enzymes. And a very interesting aspect is immunogenic, immune modulating structures. If we want to develop vaccines, it's also stabilizing structure for functional lipid membranes, liposomes, emulsomes, and some aspects of biomineralization, which I will describe in the following. I move now I extend this molecular construction kit by adding the lipids into the system. So it's a biomimetic cell membrane, so Sleytr supported functional lipid membranes. And at that point, I would like to start again with some basic informations. Most archaea, with very few exceptions, do have Sleytrs as exclusive oil component, what I mentioned already. This is shown here in the schematic drawing. You have the Sleytr, a hydrophobic domain. This can be a second protein in some organisms which are sort of linked in the hydrophobic part, and there might be functionalities. And this is just a thin section. But what we found by different methods, such a system generates a nano-pattern fluidity of the lipid membrane. And this might be the reason why these organisms can grow, some of them, under extreme environmental conditions, like 120 degrees centigrade, pHO, a concentrated salt solution, or even at high pressure. And we thought if nature has invented or found such a solution, why not apply that for nanobiotechnological applications? And we called it Sleytr supported lipid membranes, so the same fluid lipid membrane. We studied that by sort of crystallizing Sleytrs on spread lipid films. Such system can be is stable enough to spread porous structures, like you can see here. We know this all from different biophysical data. What you see here is that we also can incorporate in such a system. In this case, the Sleytrs in different color, but we have this heptameric pore from the alpha hemolycine, which is one of the best studied pore forming proteins. Now, and that leads us to a three-dimensional structure, which we call biomimetic viruses or virus-like particles. And this is Sleytr coated liposomes and emulsomes. Again, we just copy nature. Most animal and human viruses are coated with a piece of membrane derived from the cell which produces the virus. We have this in the HIV virus or with the SARS COVID-19 now problem. The Sleytr are coated with this Sleytr. We see this in freeze etching. And now we can sort of working with the system I've described before. We can have lipid vesicles. We can incorporate functional molecules. In such liposomes, there are thousands of papers describing how to entrap water-soluble molecules or DNA. And we can use the native Sleytr and this also the Sleytr, which has some functionality on the surface. The same applies to emulsomes. In this case, we have the possibility for dissolving before that or entrapping hydrophobic parts. This is just the system we are studying in the presently. This is the Sleytr fusion protein using parts of the spike protein, SARS-CoV-2 spike protein. This is the receptor binding domain or the receptor binding motif linked to an Sleytr from a bacillus using a flexible linker. Now, this gives you just an idea that such systems, this in this case are emulsomes, are endocytosis, can occur observed in cells. These are HeLa cells, and we have in this case also attached a plasmid encoding for green fluorescence protein, and here we see that this transfection system really works. This is just another example. Here we entrapped ferritin because in a liposome, because ferritin is easily recognized in thin sections due to the strong electron scattering power. And here we have the ferritin taken up by HepG2 cells. So acellular liposomes and acellular emulsomes, what is new in comparison to naked liposomes and emulsomes? We have a much higher mechanical and thermal stability. I can't describe details here, but we also have the possibility for cross-linking the acellular protein. And what we found is that using the right cross-linking, intra and intermolecular cross-linking, even the functionality on the S layer is maintained. Then we have a monomolecular coating with functional molecules, addressor molecules. We have the possibility for transporting hydrophilic and hydrophobic substances. We have here some very interesting results with curcumin, by the way. And the potential applications, yeah, this is already shown in schematic drawing before. We included as vaccines, immunotherapy, drug targeting, drug delivery, and what we call artificial viruses. So in my last slide, I will give some future aspects, and this is acellular stabilized extracellular vesicles. So these are sort of extracellular vesicles comprise a radically new idea for therapeutic treatment and prophylactic functions. Interesting 3D communication structures, and they are unexplored therapeutic and prophylactic values. There are many applications for that, and precise protocols also for isolating certain types of these vesicles. And here one can also use these magnetic beads, and acellular proteins are compatible materials for stabilizing, functionalizing, and labeling of cellular vesicles. I thank you for your attention.
Thank you very much for listening to my talk. Thanks, Dr. David Jean and Dr. Bernard Siegel for inviting me to give a talk at the World Stem Cell Summit 2021. And my talk will be focused on protein design. And the master design in the world is Leonardo da Vinci. He said more than 500 years ago, simplicity is the ultimate sophistication. Design requires you know everything from bottom up, every single element. While in the era of modern biology, and it started by the four icons here. This is Francis Crick, this is James Watson, and between them, then discovered the DNA double helix structure. Here is Leslie Ogle. Here is Alexander Rich, my mentor since 1988. This side is alpha helix. It's a protein alpha helix. It's not DNA double helix, hanging on top of the house of Francis Crick's home in Cambridge, England, in the 1920 Portugal place. And Alexander Rich has a lot of wisdom. He said to me numerous times, I benefited his wisdom greatly. When in 2003, I decided to change my research direction, and I seeking his advice. He said, it's important to know what to do. It is equally important to know what not to do. And he also said to select good project and complete it in a reasonable amount of time. Finally, he said, persistence is luck. How true it is. My title of the talk is Curiosity-Driven Research, the QTY Code for protein design. I'm from MIT, the Media Lab. I call my laboratory of molecular structure, molecular architecture. Here is a Media Lab. IMP designed the building. When he designed in 1980s, he designed MIT into the building. Here is letter M, here is letter I, here is a T. So architecture and molecular structure share a lot in common. And protein structure and DNA structure are also architecture and molecular scale. And here are the people who work on the QTY code with me for the last 10 years. Dr. Karina Koren, Dr. Loda Tickler, visiting professor Tang Hongzhi, visiting professor Tao Fei, and postdocs Han Qiuyu, and research scientist Qin Rui, and MIT graduate student Michael Skursky, and visiting professor Hao Silei. So this slide explains the importance of codes. And it's well known, the Morse code that generated billions of dollars in last century, 100 years ago, through a very simple code, and effectively transformed the human communication at a long distance. The computer code is simple code, is 0 and 1, but can transform essentially human civilization, and in the meantime, generate millions or trillions economy in the world through a very simple code. The genetic code is the oldest code, and in biology, the entire living system on the planet shares the same genetic code. That's the reason viruses and bacteria can infect people, and then coronavirus can infect people, because coronavirus use exactly the same genetic code as a human. Finally, it's my QTY code, my invention in 2011. So this code allows protein design to change protein from water-insoluble version into water-soluble version. So this talk, I will give you more in detail. Here is the example, water-insoluble protein detergent, and once become water-soluble, no detergent is needed. The protein can be soluble in water. So this I will explain more in detail here. One of the most well-known protein in human and in all the living mammals and birds and so on is called hemoglobin. Hemoglobin carries oxygen, transport, and carbon dioxide. So hemoglobin is the most well-known water-soluble protein in the protein family. It can solubilize in human red blood cells for about 30% and the protein in the cell. That's a tremendous amount of protein in the cell. The person who determined the structure in 1959 and published paper, he spent 27 years working on structure, and Max Brut, he won a Nobel Prize in chemistry in 1962. The same year, Watson and Crick won the Nobel Prize in medicine. So hemoglobin is a tetramer protein. It's more than 80%. It's alpha helices. You can see it's a helices, but it's not double helix, it's single helix. It's hanging on the Francis Crick's house. There's another protein, it's called Potassium Channel. This protein, it also can see most alpha helices. It's also tetramer. It's similar in structure like the hemoglobin. However, this protein is a membrane protein. This protein is totally insoluble in water system. But you would say how one protein is most alpha helix is water soluble. The other protein similar as alpha helix, but it's water insoluble. That I will explain. So in structural biology, also in biology, and there are three types protein alpha helices. So first on the left is protein alpha helix. It's mostly called hydrophobic residues. That is the water insoluble amino acids, and they're labeled with green. The second, the alpha helix is a half. Here is water insoluble amino acids. The other half is water soluble amino acids, labeled blue and red, pink here. And the third one is this protein alpha helix. It's mostly water soluble amino acid residues. If you can see, all the numbers here are quite different. However, those proteins share identical backbone structure. For example, they all have the same 1.5 angstrom per amino acid rise, 100 degree per amino acid turn, 3.6 amino acid, 306 degrees, or 5.4 angstrom per helical turn. Furthermore, there's another fact in protein structure. That is the 20 amino acids showing here. This amino acid listed is by size, okay, not by its chemical nature, but by size. So smallest on the left upper corner, the largest on the right bottom corner here. So smallest one is called glycine, and then increase by size, alanine, serine, cysteine, and so on and so forth. But you can see immediately there's something striking and then showing here. So these two amino acids here, one is water insoluble valine here, and totally water insoluble, putting water. The other one is called the spirulina, is water soluble. If you look at the density map here, they're nearly identical. This one pair. The other pair is leucine here, and the leucine and is water insoluble, and this is aspartic acid. Look at the density map. Again, it's very similar. And this asparagine, this is one blue, one red, and also the two more similar to leucine. That is one's glutamate with two oxygen here, and one glutamine with nitrogen here, oxygen here. This asparagine and glutamine with blue here, those have bear no charges in protein. But the other one too is aspartic acid, amino acids, and glutamate with two red, and bear negative charge. And if you're looking down here, large amino acids here. So this is called phenylalanine. Phenylalanine is also water insoluble amino acids. But the two of them are very similar to this. One is called histidine, and the two blue here, it bear positive charge, and negative, this tyrosine. The tyrosine has a red here, but that's bear no charge, just like alcohol and just OH group here. So this pair is also very similar. Only difference between those two is one extra OH group here. So this is one pair. This is another pair here, or one with similar amino acids, and this is a third pair. So this molecule here is amino acid called Q, glutamine, And this is called threonine, this is called tyrosine. So I use those 3 amino acids to replace the water-insoluble valine and leucine, isoleucine, and phenylalanine. So I call those system QTY code. So how the amino acid dissolve in water, shown here. So this is side chain of amino acids. And there's the red one is oxygen interact with the 2 molecule water. The blue one is nitrogen and also interact 2 molecule water, but they're different. In this case, these 2 hydrogen as a donor. In this case, hydrogen, the oxygen as a acceptor for these 2 water interact. It's glutamine or asparagine. The other is a serine, threonine, or tyrosine with OH group on the other side chain. This molecule can interact with 3 additional water molecules here. The hydrogen interact with the water, this as a donor, and this oxygen as an acceptor 2 more water molecules. So this amino acid, those amino acids here will become water soluble. So our idea is, and to take the membrane protein labeled here, the green, and to remove them from membrane, is the lipids, the total water soluble, and convert them, take them out, and this, without the change, then we'll need the detergent to stabilize them in outside the cell membrane. But our idea is take them out from cell membrane and directly soluble in water from here to here. And so this is very important. We want to do a demo here. So think of the surgical gloves. Okay, so the surgical gloves is hydrophobic. They can protect your hands from interact with water. So in this case, will be membrane protein. Folded, my finger folded into 5 finger here, similar like this membrane protein. Okay, it can has and function to signal transduction, for example. And in previous case, people have shown, other scientists at UC San Francisco shown this protein, this peptide here, and is hydrophobic, showed labeling green. And the crystal structure of those 2 are nearly identical. So that's green, water insoluble version, and this is a water soluble version of a conversion from this one. So now become water soluble, change 6 amino acids out of 25, 24% change. It become, the protein now become water soluble. 24% change is substantial. And we are inspired by that work. We also design membrane protein, making them to become water soluble. So after 7 years, persistent is luck, the wisdom of Alexander Rich, we finally conquered this research, this make membrane protein become water soluble. So I call this QTY code, enable design detergent-free chemokine receptor that retain ligand binding activities. Here is the example. The leucine is water insoluble and can be converted into asparagine, water soluble, or into Q is glutamine. Isoleucine or valine convert into threonine, water insoluble version, and water soluble version. Finally, phenylalanine, water insoluble, can be converted into a water soluble tyrosine. Here is a single alpha helix. Look at mostly green. The green means water insoluble. We apply QTY code. Now, after this, the alpha helix become water soluble. In analogy I showed you earlier, this water insoluble system hydrophobic. Now, once you take it off, this becomes water-soluble version. So the function has not changed. You still have 5 fingers, and you can still perform functions without hindrance. That's how surgical doctors in the surgical room wear the gloves and did not hindrance operating. And it's human for operations. So here is an example of the 4 protein we have carried out to convert from water-insoluble version into water-soluble variant. And if you look carefully, this is protein here we call CSR5 and CSR2 QTY. The number here change is very minimal. There's a PI 9.21 change to 9.10. The change is 0.1 pH unit. And the molecular weight is 40.52 or 40.98. Again, change is minimal. However, we change very large number of membrane protein hydrophobic amino acid residues. And in the transmembrane domain, we changed and it's 46.7%. And overall protein change is 21.88%, which is substantial change. Okay, if you look at this one, called C6R7, here it's PI, here is 7.52, and the native structure, native protein, and after change, PI 7.50. The PI change is 0.02. It's shown negligible, PI change. So that means and the protein structure will not change, and the function will not likely change. And also look molecular weight change, and it's 41.49 and 41.96, about 500 Daltons change. But if you change, look at transmembrane domain change is 56%, and the overall change is 23%. So there's 4 examples such change. We have now, that was initially carried out up to 2018. We have now done more than a dozen proteins in similar nature. There's another example here. So this is the CSR5 7.99 and 7.95. The change 0.04 change, very little change. Yeah, but the overall transmembrane domain change is 53%, and the overall transmembrane and the overall protein change is almost 24%. However, if you look at the structure, and the structure, the pink one, the magenta color, is a native protein. And the teal color is the QTY version. And you can see, then more or less holds the structure. This looking from the front, this looking from the back, this looking from the top. So that means the protein, although we change huge numbers, and then in the transmembrane domain, in the overall protein change, but the protein still holds a similar structure, thus has similar function. And here, showing example here. So those 4 proteins here, and it's called a native protein, then have 7 transmembrane domains, 1, 2, 3, 4, 5, 6, 7. This is 6.5. It's almost 7. So that's before change into QTY form. Those proteins are totally water insoluble. But once you apply the QTY code, all the water insoluble segment disappear and become soluble. As I said earlier, and it's like a glove. Before you, it's this glove. That's before it's on the left side, and then it's water insoluble, which but function has not changed. And on the right side, essentially take the gloves off, and the hand become water soluble system, and it did not deter any function of the hand. So the key is to understand if once you change, can you still bind something? The answer is yes. Just like with my gloves, I can hold the object without a change. I can hold object without a change. Okay, so with gloves, without gloves, they have no change for the function. So this is experimental demonstration that you change from water insoluble version to water soluble version, then more or less did not change its function. So I want to show you again. So once you will put the gloves on, you can hold it the same way, and you can hold the same way without a change. So this experiment is a very detailed scientific experiment called the ligand binding, and essentially did not change. That is, the surgical gloves did not deter the surgeons to hold the knife to the surgical operations. Okay. So the QTY code is analogous to DNA code. Okay, DNA is well known. You call it T paired with A, C paired with G, A paired with T, G paired with C. So this direction is called bidirectional. Once you know TC, AG, you immediately write out AG, TC. So it's bidirectional. So I believe the QTY code is similar. So there's a A paired with a T, G paired with a C. The QTY code is also reversible. The Q can pair with L, and the T can change V, replace V and I, which is isoleucine. The Y replace F. And it's also bidirectional, can also be L change QTY into QTY replace L, but L can also replace Q. So that's a very powerful, simplest code that Da Vinci said, simplicity is ultimate sophistication, as we know in the case of DNA, in the case of QTY code protein. Since 2018, we published 5 papers, and the first one was published in 2018. This was published in 2019, and Dr. Qin Rui is the first author, and with other people and helping doing experiments. So in this case, a QTY code designed thermostable and water-soluble chimeric chemical receptor with tunable ligand affinity, essentially doing protein design from give you some base and design some new variants. And Qin Rui also studied your same QTY code and studied non-full-length water-soluble 6L4 QTY and 6L5 QTY chemical receptors, implication for overlooked truncated but functional membrane receptors. So this paper was published in 2020. And Dr. Carolina Coren and Dr. Loda Tegler and published a paper in Scientific Report in 2020. The G protein coupled receptors 6L4 designed by the QTY code become more hydrophilic and retains cell signaling activity. Okay, that suggests there's a protein you can change it to completely the water-soluble version or change it to only partial water-soluble version. But if you change it only partial, you retain the cell signaling activity. And that means your mobile phone, you change a part of them, it still works. Here, paper also published in 2020, and then I showed QTY code designed water-soluble FC fusion cytokine receptors bind to common respective ligands. So this is to combat the cytokine storm in during the COVID infection and sepsis infection and then some cancer treatment. So this paper probably suggests the QTY code is very useful, okay, for making new product for pharmaceutical industry. The question is, would the QTY code be useful? So I believe it is. So QTY code will likely have significant implications for designing water-insoluble proteins, not only for GPCRs and other membrane proteins, but also for many other water-insoluble and aggregate proteins, including beta-amyloid peptides found in Alzheimer's disease, alpha-synuclein in Parkinson's disease, island amyloid polypeptides, beta-2 microglobulin, medin, calcitonin, serum amyloid A, and therapeutic monoclonal antibodies and others. So any water-insoluble aggregate protein perhaps can be used QTY code to make them water-soluble. As you say, maybe fibrosis disease and cirrhosis, liver cirrhosis, and eye, the cataracts, that often result in the protein aggregation and other disease. So protein aggregation causes many disease. Okay, so here are the example, and then we designed the QTY code, design water-soluble and membrane proteins. Here, this is an antibody. It's called IgG. Here, example. So we now instead of put the antibody FC domain, we put the receptor onto FC domain. They keep the FC domain intact. The FAB domain is replaced by the receptor. This is one example, another example, and there's different receptors all follow example. Now the receptor become water soluble, then actually become therapeutic agent. As Professor Uwe Sleytr has shown, after you make this protein soluble, now then can also be useful for design new sensing device. This sensor has very high density in per square centimeter, and it will be useful to detect numerous diseases or environmental hazard, and then air pollution and water pollution and so on. Okay, so follow Alexander Rich's advice. He said it's important to know what to do. It's equally important to know what not to do, and persistence is luck. And indeed, the entire research was started from a question by Alexander Rich. He asked me, can you convert water-insoluble alpha helix in the water-soluble one? And that question stimulated me to pursue this research, and it's in the last 7 years. Thank you very much. Bye-bye.